I hereby open this academic session in which candidate Xi Yang will defend the academic thesis Brain Plasticity Across the Lifespan, New Approaches with Structural and Functional Neuroimaging. Xi Yang,
yes.
Might I invite you to present a summary of your work for us here?
Thank you Thank you for your introduction, Rector. And then I will give a summary on my studies. And okay, I will start. Dear Rector, the member of doctoral community, friends and colleagues, I'm very glad you can join my PhD defenses today. In the next 15 minutes, I would like to share my work in the last few years. And my thesis is entitled Brain Plasticity Across the Lifespan, New Approach with Structure and Function Neuroimaging. What is brain plasticity? Brain plasticity refers to the brain's intrinsic ability to reorganize its function and structure in response to stimuli and injury both from the internal and external sources. And the brain plasticity mainly includes two types. The first one is structural plasticity. The structural plasticity involves the physical changes in brain architecture that can be measured by the structural MRI. And the function plasticity encompasses the brain's ability to reconfigure its activity and a connection between the different brain network or brain regions that can be measured by the function MRI and FNIRS technique. So the aim of this study is trying to use those multiple modern neuroimaging techniques, including the structure and function MRI and FNIRS, to explore the brain structure and function plasticity in the different individuals from across the lifespan. This work transcends the limitation of the traditional single-modality neuroimaging technique and provides us a comprehensive understanding of brain plasticity. By using those modern neuroimaging techniques, we can investigate the brain plasticity in a series of methodological frameworks, including the neural enhancement in the healthy participant, and the neural compensation mechanism in the old participant, and the neural interaction in the psychosis, risk, and transition group. And then I will give you the details of each study. Let's start with the neural enhancement. Neural enhancement refers to the intervention designed to improve brain function and cognitive ability. So in the first two studies, we want to explore the robustness of neural feedback training on the cortical and subcortical region. So in the first study, we want to know if the healthy participant can learn to upregulate the mesolimbic network activity. At first, we use a monetary incentive delay task to identify our region of interest for the subsequent neural feedback training. And then participants received 3 sessions of neural feedback training in 3 weeks. Participants were asked to regulate their brain activity into the low and high level. The participants were also informed that the gray bar in the thermometer represents their target area brain activity in the real time. And the green arrow indicates the target neural feedback, target low and high neural feedback condition. Our result shows that the healthy part, we can use monetary incentive data to successfully identify region of interest for each participant. And for the neurofeedback result, we can see that the healthy participant can successfully learn to upregulate their VTA activity in the first training session. But this training effect is not maintained in the next two sessions. The results suggest that even the initial VTA brain activity regulation is achievable. It also highlights the necessity to use the function localizer task in each session to identify region of interest for each session. Compared to the fMRI neurofeedback training, the FNIRS neurofeedback training is cost-efficient, practical, and even it can only detect our surface area brain activity. So in the sixth study, we want to know if the healthy participant can learn to upregulate the cortical DLPFC activity by NIRS neurofeedback training. We also want to know the transfer effect of the NIRS neurofeedback training to the related working memory task. As we can see in this slide, the participant will ask lift the stone on the beach as higher as possible to regulate the cortical DLPFC activity. And after the neurofeedback training, the participant complete the working memory task in the fMRI scanner. Our result shows that the participant can learn to upregulate the cortical DLPFC activities by NIRS neurofeedback training. And the transfer effect of the NIRS neurofeedback training shows that the neurofeedback training group increase the bilateral DLPFC activities and also decrease the function connection between the left DLPFC and the left atrial insulin. The successful upregulate the DLPFC activity and the lasting effect on the related working memory neural activity suggests that the NIRS neurofeedback training may be a promising intervention for the mental-related disorders, but this needs to be tested in the future study. The brain plasticity changes in the whole lifespan. As brain aging, the old participant may use the alternative strategy to keep the cognitive ability, such as the neural compensation strategy. The neural compensation refers the brain adaptive ability to reorganize and recreate the alternative neural pathway to maintain the function when the primary systems are impaired. So in the third study, we want to investigate the neural brain network neural compensation mechanism in the old participant. We investigate the dynamic age-related dynamic function connection change by using the open-sourced resting fMRI data from China and Germany. By using this cross-cultural replication method, we successfully found two specific brain states. The first one is the within network connection state, and the second one is the between network connection state. And for the dynamic function connection result shows that the old participant spend less time and lower occurrence in the within network connection state, but they spend more time and higher occurrence in the between network connection state. Those results may suggest that this dynamic function connection neural compensation mechanism may be a biomarker for the brain aging. In the last three chapters, we have described the brain plasticity in the old healthy participant and healthy old participant. As the brain plasticity can be go wrong in psychosis, but the underlying mechanism still not well understand. The neural interaction will provide a potential explanation. Neural interaction refers the bidirectional influence between the different neural systems to shape the overall neural organization, such as the neurotransmitter can influence the brain structure and function. So, in the last chapter, we want to investigate the neural interaction between the neural transmitter and the brain structure change in the psychosis group. The clinical high risk for psychosis are associated with widespread cortical. Here, we combine the large pooled clinical high risk for psychosis EGMA dataset with open source patch molecular map to identify if and how the neurotransmitter influence the cortical zygotes change in the psychosis risk and transition group. We found that the local neurotransmitters are significantly associated with cortical zygotes change in the psychosis risk and transition group, and the dopamine, GABA, glutamate, serotonin, norepinephrine are the most significant contributor to this cortical zygote in the psychosis risk and transition group. And additionally, we still found some risk contributors, such as histamine opioid receptors. Those results may help us to develop novel therapy in the future psychosis risk participants, but we need the further study in the future. So overall, in our work, we are trying to combine the different modern neuroimaging techniques to explore the brain plasticity across the different populations in the whole lifespan. And we found that the healthy participant cannot successfully upregulate the subcortical VTA activity across the session. But the healthy participant can learn to upregulate the cortical DLPC activity by loss neurofeedback training. And for the old participant, we found the old participants spend less time and lower occurrence in the within network connection state, but they spend more time and higher occurrence in the between network connection state. And for the psychosis group, we found that the neurotransmitters are significantly associated with structure change in the psychosis risk and transition group. That's all my presentation. Thanks for your attention. Now I would like to give word back to the director. Thank you.
Thank you very much for your presentation and the beautiful images with that. In a few seconds, we're going to test your brain plasticity, but before we do that, I'd like to introduce your promotion team. The paralympians, you can sit down, because otherwise you have to stand for 3/4 of an hour, so please take a seat. It's my pleasure to introduce to you at home and here in the audience Professor David Linden, who is a professor in translational neuroscience, Professor Ben Becker, who is all the way from Hong Kong on screen. Welcome. And also another co-promoter, Dr. Dennis Hernaus. He is an assistant professor in cognitive neuroscience. Welcome. And as you might notice, they are not allowed to ask questions anymore because they asked enough questions in the last few years. So it's my great pleasure to introduce the chair of the reading committee, Professor Koen Scheurs, with expertise in mechanisms of affective disorders at the Department of Psychiatry and Neuropsychology of Maastricht University. The floor is yours.
Thank you, Mr. Prorector. Dear candidate, first of all, I would like to congratulate you with your very nice thesis. I think it's a series of studies of very advanced level. I think I admire the work that you have done. You've clearly put a lot of effort in this. And so my congratulations, not just to you, but also to your supervisory team here and in Hong Kong. Also, by the way, congratulations with your presentation, which was visually, I think, very appealing. I appreciate that. I would like to discuss with you on Chapter 5. I have a few questions. First question is, you did a series of analyses. You used data on cortical thickness from Enigma data. You used data on PET data from an existing database on cytoarchitecture, and you used clinical data. And then could you perhaps talk me through the steps that you did to reach your end product that is presented in that chapter?
Yeah, highly estimated tolerance. Thanks for your question. So if I understand correctly, you want to give a description of my method used to analyze this data.
The steps of analysis, exactly, yes.
Yes. We use a multilinear model to identify if the local neurotransmitter can associated with the cortical thickness change in the psychosis risk group. And first we compare the cortical thickness change. Psychosis risk cortical thickness changes with the healthy participant, and we got the quantity of the whole brain differences of the psychosis compared to healthy participant, and then also for the PET molecular maps, we also have the whole brain, the activity in the whole brain petromolecular in the each receptor and transmitter. And then we combine this, use the multiple linear model to see if they are significantly associated. And then we use the dominance analysis to see the relative contribution of each contributor to see which one is more important or which one is less contributor. And we use this method. The first is got the linear model, and then is a multi-linear model to get and then is a dominance analysis to see the relative contribution. That is the flow work on this chapter.
Okay, thank you. And then you mentioned in your thesis here, and you just said as well, you used a whole brain approach, but I see a mentioning of Roy's as well. So what was the influence? How did I was a little bit confused. Was it a whole brain approach, or did you use regions of interest? And if so, which and how?
We use the whole brain, not the region of interest, because the multiple linear model, we use the neurotransmitter as an independent variable.
I'm talking about the cortical thickness now.
Yes.
When I read the bit about the cortical thickness, I was wondering, did you use any regions of interest there as well or not? Was I confused?
No, okay. Not a region. Because we have the variable limitation for the multiple linear model analysis. Because we have 80 neurotransmitter. If we want to, if the if the dependent variable more than the independent variable, we can do the multiple linear analysis. We also thought about this. And first, we want to do some subcortical analysis, but we don't have enough variable to do this, so we cannot do the ROI analysis on specific brain region. So only for the whole brain.
Okay.
Yeah.
Yes. And regarding these...
Do you have 1 second? I'm adjusting the microphone because it's too ploppy.
Okay, yeah, sure.
Stand so close to the microphone because then it pops like this.
Okay. I see. Thank you.
So, you okay? I had a question regarding these PET maps. They're from a paper from Ms. Hansen.
Yes.
I didn't know the paper, very nice paper, Nature Neuroscience. I wasn't aware, but I looked it up, and I had the impression when I looked at, there's pictures of these distribution of these different neurotransmitters in different parts of the brain. And when I look at the neurotransmitters that are most important for psychosis, the maps looked quite similar to me at first sight. So I was wondering if there are not very much differences between these neurotransmitter systems regarding their distribution in the brain. Does it make sense to map them on cortical thickness?
Because I didn't do the correlation analysis for each pattern maps, so I can't answer you if they are highly correlated. But based on the primary work, they said that the neurotransmitter distribution is highly correlated with the brain structure and function in the both healthy and psychosis patient. I think those neurotransmitters are highly correlated in this study. We use them. Yeah, I think, but I'm not pretty sure. I didn't.
Yeah, but are you now talking about correlations between the neurotransmitters and the cortical thickness? Because I was referring to the distribution of the neurotransmitters. And let's say when you look at the dopamine map, and you look at the serotonin map, you look at other maps, they look pretty much the same to me. Do you know where I'm getting at?
Yes, clear. Maybe they are quite similar, I have to admit, but I didn't. I didn't know if this seminar will influence our result. That is also maybe a limitation of my study. I have to admit that. Yeah.
Okay, no problem.
Mm-hmm.
And then I wonder if you look at, I was wondering, okay, you have now measures on cortical thickness, you have measures on cytoarchitecture. But I'm a clinician, and I would like to know then what is the chain of events? What happens in the brain according to you? Which is first? Is it first neurotransmitter changes that have an effect on cortical thickness, and then in some way on psychosis conversion? Or what's the order of things? Because that is not something that you can directly let's say, deduct from your studies. This is not a one longitudinal study. You associate different data sets. But from that, do you have an idea? What is the order of things? How does it go about in the brain?
I think I have discussed in the discussing part for this part, we cannot make a conclusion about the sequence, but we have two hypotheses. Mm-hmm. And the first one is that maybe the neurotransmitter drives the cortical thickness change neurotransmitter change first and driving the brain structure change in the psychosis. Then is based on some medicine study that if we participants take some medicine, they will on some specific neurotransmitter, and that will influence the brain structure in those patients. And also some gene studies, in some animal studies, they knock out the such as serotonin gene, and they also change the cortical thickness, in some animal. And but this is one hypothesis, but we also think that maybe the neurotransmitter and the cortical thickness change happen at the same time, because maybe some gene that control both the neurotransmitter change and also control the cortical development in the whole process. So I think we can't make a specific conclusion. We can only give a hypothesis. I think first is the neurotransmitter change. Also, maybe the neurotransmitter and the cortical thickness change happen at the same time, as those brain neurochemistry or brain architecture change and cause some positive or negative symptom for this patient. I think that's my answer. Thank you.
Okay. Yes. Clear. Thank you. And then perhaps, I'm a medical doctor. I'm a clinician. And for example, when I go to my clinic tomorrow and I talk to my colleagues and I tell about this brilliant young lady who explained her thesis. What's the takeaway message? What should I tell them? What if I see a patient before me, or if they see a patient before them with psychosis or psychosis risk? What would you advise me to tell them as what's the news in a practical sense?
Practice. So maybe I have mentioned in my slides we can maybe in the future study we can develop some therapy that combine the serotonin and the dopamine and the GABA and to this collective medication method to give the patient to avoid the further development for those psychosis risk patients. But I can't, from our result, I can't make a conclusion, because we also see some papers that they do this medication therapy on the psychosis risk patient. They also maybe have some bad influence on the cortical thickness. So maybe it's we point out a promising direction, but we need the further study to validate this result. We cannot make a conclusion about this. Just some suggestion. Thank you.
Okay, so maybe not tomorrow, but next year or so.
I hope.
Okay, thank you very much. I give the word back to the prorector.
Thank you very much. The opposition will be continued by Dr. Lydia Heltrong, expert in computational neuroscience from the University of Zurich. So, welcome to this session all the way from Zurich. And the floor is yours.
Thank you. And also from my side, congrats to your thesis. I hear an echo. Okay, it's getting better now. Congrats to your thesis, and also thanks for this nice presentation. So I would like to start my questions with a question about your pilot study with the neurofeedback on the VTA. So you use the monetary incentive delay task, the MID task, to functionally localize your VTA. But given this task, you could also analyze, or you could also calculate a reward sensitivity as a trait parameter for each participant. So my question would be, did you perform any analysis on this individual trait? So because given a reward sensitivity, participants might behave differently during the neurofeedback?
Oh, frozen. Oh, highly estimate opponent. Thank you for your question. If I understand well, you asked me for if the function localizer task can identify region of interest for each participant. I don't know if I can understand correctly. So, can you? Sorry.
Yeah. Okay. So you use the MID task to functionally localize your reward region or your target region. But given this task, you could also calculate a performance measure for each participant about their sensitivity towards a reward. And this might enhance the overall project. So my idea was whether you did any additional analysis that is not in your thesis, given such a reward sensitivity.
I didn't do the further analysis for the localizer task. We just do the complete the. We just do the primary analysis to make sure we can active the VTA activity. And for the performance in this task, I didn't see those results until now. And I think in the next step, I will continue to do the further studies, just like the behavioral and neural activation in interaction analysis in the next step. But I didn't do this now. This is just our primary result. Thanks for your question.
Okay, yeah, and my second question would be then what you said now. So if we imagine you get a big funding and you can continue to study and perform a larger cohort of this. So what would be your extensions to the study? What would you make differently? So how would you perform now the next study if you would have the money for it?
I think thanks for your question. I think in the next step, just as we have said in the thesis, the first that we should do the localizer task in each session, and to make sure we can localize the VTA in each session, because maybe only based on the first session, the localizer task after participant go out from the scanner, the next session, maybe the localization is not very correctly. And we also have some further further suggestion on what we will do. Maybe we should give some specific advice when they do the task, because some participants report that they found that at the beginning of the training, maybe some reward imagine tasks are a lot of work, and they switch the strategy to think something very suddenly. I think maybe this, because we don't limit the strategy they have used in the whole session, maybe also influence our results. So if I will continue to do this study, we will give some specific strategy to regulate the brain activity, especially some strategy related about the reward or happiness or good things, I think. Thank you.
Thanks to you. And then the second study I would like to ask you about is your FNIRS study, where you performed the training over the DLPFC. So also starting with a technical question. So you had a hypothesis about the DLPFC, and you used 8 channels placed over the DLPFC. But my question would be, why did you exclude like the rest of the brain? What was the reasoning to really just focus there and not looking for effects with other channels?
Thanks for your question. We not only see the tuning effect on our target channel, we also see some effect on the some channel close to our target channel. Just like in our study, we use the channel 6 as our target channel. This is based on our pilot study on the 6 participants. And we can find that this channel is most active during the tuning. And then we use this most active tuning as our target channel, and at the same time we can see that some channels surround this target channel also active during the neural feedback tuning. So we also see the effect of those channels, and we can also find that those related channels also increase the brain activity during the tuning, but it's not strong enough as our target channel. And because we found a result in the left channel, left hemisphere, but we the channel in the left hemisphere, both of the channels are increase the brain activity. But for the opposite hemisphere, we don't find a very strong activity changes. So I think this, even we focus on a specific channel, we can find some activity in the close by channel. Okay, that's my answer. Thank you.
And is it then a timing issue, not using like whole brain electrodes? I'm not a NIRS expert.
Because the first, this result is based on our primary study on the OFC, and we found that the participants have very good singular in the front area, but for the back of the head, the singular is very bad. So they have very good architecture, and they very easily influenced by the movement and here. So we focus on the forehead, and also by the technical limitation, because our large equipment only have around 40 channels. We cannot cover the whole brain, so we choose the most promising area to do the tuning. Yes.
Okay, thank you. And could you also elaborate a bit?
Well, the time is almost up, Dr. Helroom. The time is almost up. So if you have a very short question, we can do that, yeah.
Very short, would you expect stronger effects for using a more difficult N-back? So like using a 5-back, would you expect then more stronger effects than you have with 2-back?
Yeah. When we do the pilot study, we try the 3-back, but the accuracy is very low. I thought it's about 17%. So I think it's not very good to detect the neural activity. So we only choose the 0-back and 2-back in my study. This is based on the pilot result to, yes. Thank you.
Okay, thank you.
Okay. Thank you very much, Dr. Helroom. Then I'm very happy to pronounce the next opponent is Professor Klaus Matiak. He has a chair in psychiatry and at the University Aachen. Welcome for taking this journey all the way to Maastricht.
Yes.
Floor is yours.
Thank you for inviting me, and thank you for this nice presentation, which I think is illustrating that even our clinical thinking nowadays is really influenced by imaging findings and the integration of different imaging, systemic measurement modalities across different domains. So, particularly, I think also that was very nice, illustrative, that we see changes in aging. I think that's also kind of the title. No, we have brain plasticity changes over the lifespan. But I think also what we learned from research into aging, it's not all bad. Yeah, so there is actually some improvements over aging, like something like static intelligence, for instance. So I wanted to ask you, when you think about modifying and neuroenhancement across lifespan, is that different? So is it different if I have a adolescence and do some training to improve brain networks compared to an elderly professor being 60 years old?
We have thought about this. Just like our group are doing some network brain neurofeedback training. Highly estimate Poland professor. We have thought about this. For the old participants, we want to, and for my future career, I want to develop some network, brain network level neurofeedback training. Just like we have seen that in our chapter 4, we found that the old participant compared the younger participant, they spend less time and lower occurrence in the within network stage. So our training, in the future training, we want to do some FMI training on the within network, just like we do some neurofeedback training within the DMN or the CN network. But for the younger participant, because I don't have some experience on the teenagers or training, we are mainly focused on the healthy young participants more than the 80 ages. But I don't know if we can do some with between network connection to improve their brain development or to improve the cognitive ability. But I want to see in the future study for the old participants, we want to do some within network connection neurofeedback training. Thank you for your question. That's my answer.
Okay. Yes. Okay. Let's stay with that, because my second question is kind of actually already in parts answered by your reply now. So, I mean, you have a nice experience looking into meta-analysis, looking into large data set. And I think, especially in the neurofeedback community, we often wonder what is the best target for neurofeedback. Yeah? And so we have these ROI approaches, as you presented, and the network approaches, what you presented. So I think a little bit you answered this already, but maybe you can also more specify where would you see application of localized neurofeedback techniques, and where would you see the main target on using network-based approaches as neurofeedback signal?
I think based on our group previous work, we use some resistance data scan to define the lateral work that we are interested in. Just like in our colleagues, they do 5 minutes resistance scan at the beginning of the neurofeedback training to choose the DMN and see lateral work, and then use this. We are not use a specific task, just some reasoning scan to localize our network, our interest in. So maybe there are some different from the task related localized task. Some study is based on the some studies they only use a reasoning scan, but there are also some studies use some cognitive task to identify the region of network. Just same as our study, some working memory task to identify the CEN and also some self related task to define our DMN network. So this we cannot make a conclusion. We should based on our specific aim and our specific participant. So that's my answer.
Yeah, but then I want to ask specifically. For instance, let's take hallucinations in schizophrenia. So you have auditory hallucination. Would that be for you an argument to address the auditory cortex, for instance, as a region? Or would you say, okay, we want to modify the network which is supporting this clinical phenomenon? Is that...
Sorry.
So what would be intuition when we go and say, okay, we have a patient with auditory verbal hallucination, so he's hearing voices in his head, and that's a very strong and limiting phenomenon and symptom. And so when you would design a neurofeedback experiment, would you go there for a region, or would you think that's really something where the whole networks have to be modified?
This is very difficult. I think for this should based on the previous literature. So just like some psychosis patients, they have the thesis on the just like the mesolimbic network. In this case, we should maybe same as our first study to use the function localized task to identify region of interest. I don't have a specific answer for this, but it's case by case. Just for psychosis, I think we can use our first paradigm to do the neurofeedback training.
Okay, but I understand your focus would be to go for the individual findings and see based on this navigator approach. Very nice, thank you. I think I still have a minute or two left. And then I think also, again, I want to direct towards more the further development of neurofeedback specific again. Yes, we have the region of interest, we have the network things, but also you showed, and I think it's nice to see also how you can integrate data from metabolic information, dopamine, serotonin, etc., and regional brain activation and psychopathology. So do you see a future there where we do neurofeedback based on spectroscopic data, so basically to directly start to modify the neurotransmitter?
Yes, you just point out what we will do maybe in this or next year. We have discussed with my supervisor team. We want to also because I think David is the chair of EGMA group. We have and my supervisor in Hong Kong is also expert in this field. We also want to combine this accessible and also some open source neurofeedback data to do some to do some studies similar like our chapter 4 to see if we want to do some meta-analysis to see if and how the neurotransmitter can influence the brain function changes during neurofeedback training. Just like from previous meta-analysis result, we can consistently show that the participants are active in the VTA and also some reward network, just like CEM and DLPFC activity. We want to do this whole brain meta-analysis to see if and how the local transmitter influence the the brain activity during the training. But this is what we will do in the next step, but we didn't reach it until now. Thank you.
Okay. Thank you very much. Then don't forget to inform me about your future findings. Yeah, thank you.
Thank you.
Thank you, Professor Matjak. Then we continue the opposition with Dr. Gabriella Blokland. She's an assistant professor in genetic imaging from the Department of Psychiatry and Neuropsychology of this university, and she's also secretary of this committee. Floor is yours.
Thank you, Pro-Rector. Dear candidate, first of all, congratulations on completing a very interesting thesis. I enjoyed reading it, and congratulations, of course, to your supervisory team. So, my first question, it kind of applies to both chapter 2 and chapter 3, which were both neurofeedback studies. And as I was reading your methods for the neurofeedback training, I had a few questions that occurred to me. So first of all, for instance, in chapter 2, you say, our study employed only general instructions and encouraged participants to develop their own strategies, which may have influenced the neurofeedback training effects and overall change in activity effect. So I was wondering, what way would you expect the instructions and the strategy to maybe have affected the neurofeedback training effects? And were participants asked about their strategy? And if yes, what did you learn? If no, why not?
Highly estimate opponent. Thanks for your question. Because we can, after the neurofeedback training of each session, the participants were asked to write down what strategy they have used. But maybe in the process of the training, they maybe change the strategy because they maybe find this did not work and they will change to next strategy. But because our training effect on the VTA is not very successful, we cannot make a conclusion what strategy is most efficient. But based on the previous literature, they do some correlation analysis about strategy used during the neurofeedback training and the VTA activity. They found the most important and most useful strategy is some positive thinking, some just like the happiness time with friend, some monetary reward, and the traveling in overboard, lie on the beach, just like those strategy. So I can see in our study we cannot make a conclusion about this, but from the previous literature, maybe the positive strategy is most useful. This is what I have said in my thesis, and maybe in the next step we can use a limited strategy, just some positive thinking. But yeah, that's my answer about this question.
So if a lot of people use this positive thinking strategy, what does that maybe have for implications for who this neurofeedback training might be successful for? So I imagine, there could be differences in characteristics of if we're thinking about patients, in characteristics of patients, and some might have more difficulty with this positive thinking than others. So how would you envision that would affect like who this works for?
You mean that maybe some patients they cannot imagine some positive? Yeah. That's a good question. I never thought about this. I think maybe we can do some pre-test to, at least they can, we can ask the participant to make a list to patient, we are focused on patient, to make a list of what the most motivated things to him. And then during the neurofeedback training, we ask them to think about those most motivated strategy. Or we can, or we can do some studies, just like my previous study, we can recruit some participant, they say they have some, they love some pets or love food, this participant, and we select those images for them to ask them to think about those images, to just happy time with their pets or think about some food to make sure they can successful to active during the neurofeedback training. But I think from our perspective, I don't have some very good strategy to control those thinking.
All right, thank you. My other question, it kind of relates to how neuroplastic changes may differ, and Professor Matiak asked about how they might differ across the lifespan. And my question is, how might they differ across the sexes? So in chapter 3 about the Effner's informed neurofeedback, you controlled for sex differences and to control for sex differences and reduce potential menstrual cycle effects, we only enrolled male participants in this study. And we know more and more now that males have variations in their hormonal cycles as well that fluctuate daily, maybe seasonally. Testosterone peaks in the morning and decreases throughout the day. And similarly, it might vary throughout the year. And they may be more subtle and more kind of continuous, but they can still affect like mood and energy and focus, libido. So I was wondering if you accounted for this in any way in your data acquisition. And in future studies, how would you, how might you address that differently? How would you expect female neuroplastic changes to vary across the menstrual cycle? And how would you handle that?
Yes. We thought about this because when we design this experiment, we just think that the women are very easily influenced by the hormone levels, such that we are during the period we are feel very uncomfortable. We cannot. Usually, we will not ask them about question if you are during the period, but. In our study, we just want to control the gender differences, so we only focus on the male study, neurofeedback training. But I think from the previous studies, they use the same platform and the same regimen. They didn't find the gender differences about the training sources. And I think maybe the gender will not influence our result, but this indeed needs the further test and validation. If I have funding support in the next year, I will also want to see if this nurse training will also work for the female participant. But we can hypothesize that maybe the training effect is same, but just a hypothesis, I have to say. We don't know the correct result. Yeah.
Perfect timing. Thank you very much for your very enthusiastic defense. As you have seen, the defense time is over now, so I will ask you to stay here together with your company while we withdraw for our discussion about the quality of your thesis and the quality of your defense. So please remain seated here. We will draw now. Thank you.
Thank you.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose that branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. The mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Dear candidate, dear Xi Yang, I'm here. Yeah. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee here has decided to grant you the degree of Doctor. Professor Linden is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
All right, please.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Xi Yang, CC, the degree of Doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the Rector, the Secretary, and the other members of the committee, and affixed with the official seal of the university.
Thank you.
Please be seated.
Great. So, Dr. Yang, so I have the honors to present the laudatio to you today.
Thank you.
Dear Dr. Yang, so today marks a well-deserved milestone in your academic journey, and it must feel really gratifying to finally hear these words. You did it.
Yes.
After 4.5 years of hard work, perseverance, and determination, it is time to formally recognize and celebrate this achievement. And we will also later today. Let us take a brief moment to return to the very beginning, because that's what the laudatio is about, right? Let's look back. It was in the spring of 2021, in the middle of a global pandemic, that you arrived here in the Netherlands. Travel restrictions, social distancing, and institutional uncertainty were still the norm, and yet here you were, ready to begin your PhD journey, unaffected by all this turbulence around you. On April 14, I don't know if you remember this, but on April 14, 2021, I received an email from you, and you were asking to meet. I have to admit, I was very surprised. I had previously seen your CV, and the brilliant Professor Linden and I had discussed the possibility of hosting your PhD, but you and I, we had never actually spoken. But suddenly you were here in the Netherlands with a fully functional email address, ready to meet, and as I would soon find out in our first meeting, ready to get started.
Yeah.
In hindsight, that moment, I think, was the first glimpse of your proactive attitude, and it was certainly not the last. In fact, I think proactivity, that's a quality that would go on to define your PhD journey. Because within 10 minutes of our first meeting, you enthusiastically shared a list of research ideas with me, an entire roadmap of what you hoped to explore. And I recall, because it was 2021, I recall that I was trying to temper your enthusiasm, because after all, we were still in the middle of a pandemic, and some things might move a little bit slowly. But slowing down was not part of your plan. To stay productive, you quickly downloaded not one, but 2 open source fMRI datasets. You applied advanced techniques, and voila, within weeks, the analyses were done. Your first chapter, your first paper was done. Then you quickly moved on to your FNURS neurofeedback project, which was, of course, under the supervision of our esteemed colleague and co-promoter, the brilliant Professor Becker, who's sitting there in the back. And when you encountered technical limitations during these projects, I remember there was a particular limitation. You had a lack of computing power. You didn't wait for bureaucratic approval. You didn't wait for administrative holdups. No, you simply ordered your own PC online, because why go slow if you can go fast, right? And as you might remember, this caused a whole bureaucratic outrage. Another great example of this was your reward neurofeedback study, right? This proactivity I've been talking about. After many delays, this came to life in late 2023, this project. And your dedication during this period was widely noticed by everyone. Your friend and colleague Xiao Wu observed your rigor and commitment. A small quote, "You attended every training session. You planned all scanning appointments with great care and made sure that every detail of the experiment ran smoothly. If there was a shortage of participants, no problem. That didn't stop you. You just go to the hallways with flyers. You just speak to strangers and recruited whoever was willing to participate." I remember the study went really fast. It was completed in a matter of months. And at times you were so immersed in this work that you even forgot to eat. A real testament to your perseverance and your responsibility. But of course, your perseverance, this has always been evident for us. After all, you moved from China all the way to our small country, a move that understandably came with its fair share of challenges. You encountered a new culture, new customs, a different rhythm of life, and of course, new food. And one of my secret sources tells me that one of the biggest cultural adjustments for you was waiting. Everything here just seemed to take a little long. I wrote this with 5 O's. In Maastricht, apparently life moves a little bit slower. Project planning might take a couple of months. Then there's training sessions spaced out over weeks. The approval process may take another couple of months. It all felt a little slow for someone as driven and efficient as you. And then there was the food. While others may be intrigued by the novelty of new tastes, I don't think the Dutch sweetness was really to your liking, right?
Spicy food.
Exactly, yeah. Spicy food, right. An absence of spicy food was really problematic for you. You did, however, take matters into your own hands, if I remember correctly. Where you could, you recreated your favorite dishes, and the Asian supermarket here in Maastricht became your trusted ally. Yes. A little taste of home, right? Not exactly the same, but somewhere approximately in the same cuisine. And it was just around the corner. Then there was another trusted ally, your little adopted friend, Autumn. Yes. A real source of inspiration and motivation. Autumn is Cece's adopted cat. Some of you might not know this. And someone who turned your house into a home. And I'm glad to hear that Autumn has already safely arrived in China and must be waiting to be reunited. And I think this year would also be a nice moment to congratulate everyone that's joining digitally, your friends, your partner who's watching the online stream. Many congrats on this great achievement. So leaving China for the first time in your life, of course, is no small feat. And to make things more difficult, the pandemic meant that for nearly 2 years, you were unable to see your friends, your loved ones, your family. And this must have been really challenging. Only near the end of your PhD, you could finally fly back. One lovely trip over the summer last year, and then a second and final trip at the beginning of this year, which was much needed. Because, yes, in the final stretch of writing everything up, you developed a persistent and painful toothache. Yeah. As your concerned supervisor, and I remember this is not so long ago, I recommended every remedy I could think of. Special toothpaste, right? I recommended Almex, I think. Painkillers. I encouraged you more than once to see a dentist. But as it turns out, you were holding out for your own trusted dentist. After several weeks of discomfort, when you finally flew back, you immediately got treatment when you came back. I think the same day, no?
Yes, same day.
And the literal email I sent to you after you came back said, it feels very relieving, referring to the remedying the toothache. So over the past 4.5 years, I've not only come to admire your scientific rigor, your work ethic, but I've also grown genuinely fond of you, CC. It's been a real pleasure to work with you. Someone who brings not only intelligence and energy to the table, but also warmth, humility, and quiet determination. That's why I'm truly happy to hear that you may soon continue your journey in academia, most likely in China, Chengdu even. Yeah. And it seems that a job offer might be already on the table, and I honestly can't think of someone that's more deserving of the position. But even as your career takes the next step, I very much hope that we will continue to collaborate, our entire team. I'm looking forward to wrapping up the Enigma paper on neurochemical drivers, something you discussed in detail today. Also, our long-promised meta-analysis on reward neurofeedback. I'm looking forward to many more ideas, papers, and messages that start with, "Dennis, I have a new plan." And so let me just end by simply saying, and I hope I pronounce this correctly, Yi Lu Shun Feng.
Yes.
Yes? Okay, good. May the wind be always at your back as you continue your way forward. I have spoken.
Thank you.
Dear Dr. Yang,
Yes.
congratulations on behalf of Maastricht University. I'm the first official on behalf of the university to congratulate you. You did a really great job. We enjoyed your presentation, we enjoyed your enthusiastic defense. It's a pity that you go back, because we like having candidates like you. You'll be forever an alumnus of this university, and we're proud of that, so you did very well. Congratulations again.
Thank you, Congrats.
Now some household messages, I could almost say. We will first stay here a bit and take a picture of you with the certificate, your paronyms and the whole team here, and in the background are two guests who are on the internet, so then you have a picture to remember that. Then we all will go to the grand staircase, where we'll take other pictures, but before that we will invite Professor Becker and Dr. Helrung to say some words to you. So it's a bit of a mixed ceremony. Then we go to the grand staircase, and after that we go for the reception area for drinks. Having said that, I conclude this session.
Thank you.